Hello and good afternoon, and welcome to this session on Let's Crack NEET UG English. Let us first check for the audio. If the audio is good, then we will get started. So let me know if this audio is okay or not, and then we will get started. The students are going to join in, I guess, little slowly today. I don't know for what reason. Taking a little longer to join. Respiration in plants is our topic, and we will be talking about Krebs cycle in detail, the energy gain, and little introduction of electron transport chain. I'm just waiting for students to join in so that we can get started. Okay, so Meera has joined. Hi Meera, Meena. Okay, hello. Are you a new student here, or have you attended earlier lectures also? Did you attend the previous class where we were talking about a basic of Krebs cycle? And all those who are joining in, they need to check whether audio is clear or not. Meera says, no ma'am, no is, oh, you're a new student. So that means you have not attended the Krebs cycle session which we had. And how come you joined so late? Because we are done with photosynthesis, respiration in plants. There are, I think, 2 more lectures left. And on 31st, we are going to have a quiz. And did you get a chance to see all those previous lectures on these 2 chapters? Because these are very, very important chapters from plant physiology. And just let me know whether this audio is clear or not. Okay, you've seen previous lectures and that's why you're attending this. Okay, so that means you understood all the previous things which we taught, which we studied here. And which class are you in? 11th, 12th, or have you taken a drop for NEET? Okay, you are in 11th class. So this is anyways class 11th chapter, and it is important from competitive point of view also and 11th class point of view also. And we will be talking about Krebs cycle in detail, and this Krebs cycle is asked like a 5-mark question in 11th class exam. And I can see other students also joining in. Hiren, good afternoon. Why are you late? Not very late, but just 3-4 minutes. All right, so let us get started with what we have planned for today, and maybe after 5-10 minutes, once we are done with oh, you had a class, okay, Hiren, no problem. I guess that's why students are joining in little late, because they're coming from one class to the next. So that's okay. Let us talk about the basics which we discuss almost every day, and that is about the platform that we are using, about myself, and like there are new students. I just had a chat with Meera, and a new student, right? Meera is a new student, so similarly there can be other new students also. So this slide is for them. I'm your biology teacher, Neela Bakore, and you can install this app because I'm taking other classes on this Unacademy learning app, the special classes, and those special classes are also free right now. So you can install this app, the Unacademy learning app, and here you will get all the benefits of the special classes. Then you can also go for subscription plans. There are two subscriptions which are going on right now. One is Plus and other is the Iconic. I'll tell you the difference between the two also. This Iconic one has been recently introduced. So there are few things which are common in both, like in Plus and Iconic. Best educators, live classes, PDFs, structured course, quizzes, etc. This is common in both. But in Iconic, there is a personal tutor who is going to help you. Then there is a planner which is designed keeping in mind your target exam. Then there are guidelines which are given by experts and test analysis. This test analysis part is the most important part whenever we are preparing for any competitive exam. The reason is when we take a test after studying a particular chapter, and suppose there is something wrong, if we are not able to answer it correctly, then what we do is we either try to mug that answer up or we just take it in a short-term thing. But suppose the same concept is asked in a slightly different language, different words, then you will not be able to attempt that one also correctly. So it is to be analyzed that why did we go wrong, where did we go wrong, what was wrong in our thought process, and that's why this test analysis is the most important thing. Now the subscription plans for the Plus and Iconic are different. For Plus you have 1 month, 3 months, 6 month, 12 and 24-month options. And if you use this code, you will be eligible for 10% discount. And this is for Iconic. So for Iconic subscription, you have 6, 12 and 24-month plan. Here also, you can avail that 10% discount. So this is something which is new, which I wanted to share with you. I think I shared it yesterday also. But 2-3 times if you see this, you understand what exactly is coming up. Hi Kalai, Kalai has also joined. So this is again the benefits, and this is what our topic is. We are on aerobic respiration, that is respiration in plants, and we have so far done few steps. So let us just write down those in short. We have talked of glycolysis in detail. Then we have talked of a link reaction, which is also known as oxidative decarboxylation. The third step is Kreb cycle. This glycolysis we have done in detail, all 10 steps, 2 phases, how many ATPs we get, everything. Link reaction also we have done in detail, and we also talked about Kreb cycle. Now after we finish Kreb cycle, then the next step is going to be electron transport chain, and then the last step, that is oxidative phosphorylation. So what we have planned for today is detailed study of Krebs cycle and introduction of electron transport chain. So this is what we have planned for today. Okay, a few more students have joined. Dimple Soni has also joined. Okay, my question is to all those students who are here with me right now, did you attend yesterday's lecture? Were you there when we talked of glycolysis, link reaction and Krebs cycle in brief? Hiren says yes. What about others? Because if you have attended yesterday's class, that means you have little base of Krebs cycle. Okay, Hiren, you attended it, I remember. Others, Kalai, Dimple, Meera is new. Kalai has also attended complete. And did you get a chance to draw that Krebs cycle at home? Did you even attempt to draw that Krebs cycle? And were you able to draw it, the complete cycle? Because in 11th class, this Krebs cycle is asked as a 5-mark question. And in competitive exam, there are questions which are asked from the Krebs cycle. Alright, let us draw this Krebs cycle in detail. The brief part we have already done. Some basic things for the new students I'm going to write down. It takes place in matrix of mitochondria. This is first. Is there any other name to this cycle? Kala is writing no. No is, I think, for that you did not try to revise or draw that cycle, I guess. Ayush has also joined. Hello, Ayush. Okay. It is also known as TCA cycle. Ayush has given the example TCA, also known as citric acid cycle. Okay, so now Ayush and Hiren, Kalai, they have given this answer. What is the full form of TCA? Tricarboxylic acid cycle. TCA stands for tricarboxylic acid. TCA or tricarboxylic acid means an acid that has 3 carboxyl groups, that is 3 COOH groups. Okay, and citric acid cycle, because this is the first stable compound which is formed in this cycle. Alright, and what does it start with? Krebs cycle. It starts with, the cycle starts with acetyl coenzyme A. And how is this acetyl coenzyme A formed? From one glucose molecule we got 2 pyruvic acids. This was the glycolysis step. Now these 2 pyruvic acids, through link reaction or oxidative decarboxylation, are converted into 2 acetyl coenzyme A, because coenzyme A is added here, carbon dioxide is given here, and in this step, right, if we are talking about glycolysis, what did we get in this step? We got 2NADH2 and we got 2 ATP. This is equal to 6 ATPs. That is 2 NADH2. What did we get in oxidative decarboxylation? Here also we got 2 NADH2. And this is again 2 into 3, that is 6 ATP worth. So this is what we have so far. 8 ATP, this is gross gain, and net gain is 2 ATP. And in case of oxidative, there will be 6 ATP gain, oxidative decarboxylation. Alright, now this acetyl coenzyme A's 2 molecules will enter Krebs cycle. Alright, so this is just the basic of Krebs cycle that we already talked of, but because now we will be talking about Krebs cycle in detail, all the steps, enzymes and everything, that's why this basic is written down here. Okay, it starts with acetyl coenzyme A accepted by oxaloacetic acid. Oxaloacetic acid is a 4-carbon compound and acetyl coenzyme A is a 2-carbon compound. And the first compound that we get is citric acid, or it is also written as citrate. We have to keep a track of carbon compounds so that we know where carbon dioxide is removed and all those things. Citric acid changes into its isomer, that is isocitrate. It is an isomer, that means this is also a 6-carbon compound. Now in this part, let us write down the name of the enzyme. We are writing the enzyme for the isomerization. So here it is called citrate isomerase, because isomerization is taking place, that is why. Now what should be the name of the enzyme here? It is called citrate synthetase. So citric acid is being synthesized, and that is why the name of the enzyme is a synthetase, citrate synthetase. Let us not forget to number these reactions, because we know it is very, very, very important. This is step number 1. This is step number 2. Okay, Ayush has written the statement which I gave you earlier yesterday. See, I know some simple formula making options, and that is how you are able to remember what is going to come next. So let us see what is the next substance, but the formula doesn't give you the names of the enzymes. So here we are also trying to add the enzymes. The next substance is C-I-NO. So that is starting from K. It is alpha ketoglutarate, or it is also known as alpha ketogluteric acid. Now this alpha ketoglutarate is a 5-carbon compound. So from isocitrate, which was a 6-carbon compound, we now have a 5-carbon compound. That means 1 carbon is given out here, and that carbon is given out in the form of carbon dioxide. That means decarboxylation is taking place. Carboxylation is addition of carbon dioxide, and decarboxylation is removal or loss of carbon dioxide. So isocitrate changes into a 5-carbon compound. Let us Write down the number of the reaction or the step. Now, if you remember, I gave you an important key or information yesterday that step number 3, 4 and 8 are redox reactions. Which steps? 3, 4 and 8. See, you have to remember these numbers. How you remember it? You can find some things related to it, like date of birth or something which can give you a hint. So you remember these numbers, okay? And in this redox reaction, NAD is going to get reduced into NADH2. This is one very important thing. So now we are on step number 3. So step number 3 is a redox reaction also. So what is happening in this? NAD gets reduced into NADH2. So here we have one NADH2, which is, we know, is going to give us 3 ATPs later on. When and how, that we will see in the next steps. Okay, next steps means we are talking of electron transport chain and oxidative phosphorylation. So in step number 3, there are 2 things which are happening. One, there is loss of carbon dioxide, that is decarboxylation. There is loss of hydrogen, that means dehydrogenase must be working. And one thing is getting oxidized, other thing is getting reduced. NAD is getting reduced and citrate is getting reduced into alpha ketoglutarate. So here, if we have to write down the name of the enzyme, then what name should be given to it? The enzyme is written as isocitrate dehydrogenase. Hydrogen has been removed from isocitrate. We can also write one more name here, and that is isocitrate decarboxylase. Are you getting these names of the enzymes that we are writing here? Anybody who has any confusion in these enzymes? The reaction itself helps us identify the name of the enzyme. So one carbon dioxide is given out, one NADH2 is produced. Any problem in these 3 steps, 1, 2 and 3? If not, then let us come to the next step. Isocitrate oxidase, yes, you can. You can call it dehydrogenase or oxidase. See, this is what happens when you understand, right? If hydrogen is removed, you call it reductase, but at the same time it is undergoing oxidation. So isocitrate oxidase also, very good. Alright, now alpha ketoglutarate, which is a 5 carbon compound, is going to change into succinyl coenzyme A. And from where is this coenzyme A coming? Coenzyme A is added into this. It is present in the matrix of mitochondria. So now here we have succinyl coenzyme A. Succinyl coenzyme A is a 4 carbon compound. That means again there is 1 carbon dioxide which is given out. 1 carbon is lost in the form of carbon dioxide. Okay, and which is this reaction number? It is reaction number 4. That means it is also a redox reaction. So here also NAD gets reduced into NADH2. That means here also we have one NADH2, we obtain one NADH2. So this is also decarboxylation reaction. This is also a reduction reaction, or that means it is redox reaction. So here that's why we have written that it is a redox reaction. And we know when there is redox, one substance gets reduced, the other substance gets oxidized. This is our step number 4. Any confusion in step number 4? And who can guess the names of the enzymes that can be used here? What is the name of the enzyme that can be used here in the 4th step? Can I call it alpha ketoglutarate decarboxylase? Can I call it alpha ketoglutarate dehydrogenase? So basically here there is one dehydrogenase and one decarboxylase. So by knowing what is happening, alpha ketoglutarate oxidase, yes. So there is removal of hydrogen, there is removal of carbon dioxide, and when we know that all these things are happening, we actually are able to write down the name of the enzymes. This is our step number 4. Okay. Now let me give you one more thing here. In this case we wrote that step number 5 is phosphorylation step, and in this we get a GTP molecule which ultimately is going to give us ATP. This is our second important thing. So now as we have come to step number 5, this is our step number 5. So in step number 5 what is happening is phosphorylation of GDP. So in this case GDP molecule, GDP is a diphosphate, takes the phosphate and is changing into GTP. Now when this GTP changes back into GDP, at that time the phosphate and the energy is taken by ADP and it gets converted into ATP. So here we have one ATP molecule synthesized, but we have to remember that ATP is not directly synthesized. It is GTP first, and then through GTP we will get ATP. So write down somewhere that in step number 5, which is a phosphorylation thing, the direct ATP gain is not taking place. It is first GTP and then it is converted into ATP. The enzyme that is used in this particular reaction is a phosphorylase, and that is called, I'm writing here, it is called thiokinase. We are using a specific color for the enzymes. So even if we write it slightly away from that arrow, you should know what is happening. So in step number 5, the enzyme used is thiokinase, and kinase we know is a phosphorylase basically. And this succinyl coenzyme A is going to change into succinate or succinic acid. Succinyl coenzyme is 4-carbon compound. Succinic acid is also 4-carbon compound. Succinyl coenzyme A has coenzyme A. Succinate is without coenzyme A. So here this coenzyme A will also be lost. This is our step number 5. Then I gave you one more point in the key, and that was about step number 6. Step number 6 is also a redox reaction, but here in this redox reaction FAD gets reduced into FADH2. So these things we have to remember whenever we are drawing Krebs cycle. So now we are coming on step number 6. So now this is our step number 6, and when you know the sentence which I gave you, it is see, I know some simple. Now next is fumarate or fumaric acid formula making options. So it is fumarate or fumaric acid, and fumarate is also a 4-carbon compound. That means now no more carbons are being lost. Okay, fumarate or fumaric acid. This is step number 6. So step number 6 is also a redox reaction, but here the only difference is FAD will get reduced into FADH2, which is again going to give us ATP. We know 1 NADH2 later on will give us 3 ATPs, and 1 FADH2 will give us 2 ATPs. This also is very important. We have to remember it, and we will understand this when we talk of electron transport chain and oxidative phosphorylation. So we are done with Hiren has written 2 ATP. Where is this 2 ATP coming from? Where is this 2 ATP? Which step are you talking about? Oh, okay, you were writing about FADH2. All right. So 1 FADH2 gives you 2 ATPs. Perfect. Now, what is the next step? The next step is it is going to change into malate. See, I know some simple formula making, and option is already there. Let us write down the step numbers so that we know what is happening where. This is step number 7. This is step number 8. So we said that there are 8 steps which are in Krebs cycle, and in 3, 4 and 8, which are redox reactions, NADH2 will be formed. 6 is also a redox reaction where FADH2 is formed, and 5 is a phosphorylation where finally we will get 1 ATP. So let us come to step number 7. In step number 7 there is only one thing, water gets added. Step number 8, and okay, important thing that all these are 4-carbon compounds, so no carbon is lost as carbon dioxide. So malate is also 4 carbon, fumarate is also 4 carbon, oxaloacetic acid is also 4 carbon. Okay, Samuel and Lokpal, they have joined now. Alright, let us talk about step number 8. What is happening in step number 8? It is a redox reaction. And in this 8th step, redox reaction, NAD will get reduced into NADH2. And the name of the enzyme, here we can write the name of the enzyme as dehydrogenase. Oxidase can also be written malate oxidase, malate dehydrogenase. Okay, and then fumarate to malate, actually the enzyme is simple addition of water, enolase type, but here we need not write any enzyme. Here what should be done? Here also it is dehydrogenase. You can call it succinate dehydrogenase, you can call it succinate oxygenase or oxidase. This is your Krebs cycle. Everything is done. All 8 steps, ATP gain, let us count it now. Or before we count, any question in this particular cycle, anything that you have not clearly understood, we can take that up. Though I gave you a brief idea of this in the previous lecture, but here we have added everything, the names of the enzymes, what is getting added, what is getting lost, everything. Any doubt? Anything that is not clear in this cycle? Decarboxylase. See, addition of carbon dioxide is called carboxylation. Removal of carbon dioxide is called decarboxylation. So the enzyme which will help in removal of carbon dioxide will be called decarboxylase. ASE word is normally used for enzyme. So it is removing carbon dioxide from isocitrate. So we are calling it isocitrate decarboxylase. Thiokinase, normally thio word is used when phosphate interchange is taking place. Kinase word is used for phosphorylation. Okay? Anything else? Okay, so now let us count the ATPs. Alright? The cycle should be drawn in this manner, and if you draw a cycle like this, you will get 5 on 5 because this is going to be asked as a 5-mark question. So if you have done everything, you've written everything, and what you have to remember is this and the sentence. What was the sentence? The sentence was, see, I know some simple formula making options. So if you are able to remember that sentence and these 3, 4, 8, 5 and 6, you are done with Krebs cycle. And what is happening in that step, that will help you write down the names of the enzymes. So this part is very easy, but for that you will have to at least practice drawing this Krebs cycle at least 2-3 times. So once you do it 2-3 times, you will be all set for this. So if we go back to this point, we said we are starting with 1 glucose, we are getting 2 pyruvic acids, we are getting 2 acetyl coenzymase. That means there will be 2 Krebs cycles. if you are starting with one glucose molecule. But if you have to just calculate the ATP gain in Krebs cycle, one Krebs cycle. So what can we do? We get, let us say, what is the gain? So gain in Krebs cycle, we get 1 NADH2, 2 NADH2 and 3 NADH2. 3 NADH2, and we know 1 NADH2 is worth 3 ATPs. So it is 3 into 3, so ultimately we will get 9 ATPs. Finally, we will get, right now we got NADH2. Then we got 1 FADH2, so 1 FADH2, we know 1 FADH2 is worth 2 ATPs, so we will get 2 ATPs later on. And let us not forget this ATP. So we got 1 GTP, which is going to give us 1 ATP. 9, 10, 11 and 12. So one Krebs cycle gives us 12 ATPs. And now we know how we are getting these 12 ATPs. 9 through NADH2, 2 through FADH2 and 1 as ATP from GTP. Okay? So gross, yes. Perfect. Hiren, you got it absolutely right. Gross ATP gain or total ATP gain is 12. But if somebody says, what is your net gain in Krebs cycle? What is going to be your answer? What is our net gain of ATP in Krebs cycle? One Krebs cycle. Then what is going to be your answer? The answer is going to be 1 ATP, because here we are getting only 1 ATP which can be used as it is. And gross or total is when later on we will get that ATP. We know we have a check which is going to be encashed, and that is why we are counting it. See, I gave you an example earlier that suppose somebody gifts you, say, checks. Somebody gives you like 100 rupees check, somebody gives you 500 rupees check, and somebody gives you, say, 100 rupees. So you have two amounts in check, 100 and 500, and the cash amount is 100 rupees. So after you get the gift, how are you going to feel that you have 500 plus 100, 600 plus 100, 700? So you will feel good that you have 600. But can you spend it? You cannot. You spend only 100. But you are happy because you know that those checks are going to be encashed later on. Here also the situation is same. We don't have those 3 ATPs from NADH2. We don't have those 2 ATPs from FADH2. But we know that we will encash those checks later on. And that's why we are just counting. So that we feel good that yes, Krebs cycle is going to give us 12 ATPs. Alright, so Hiren, Kalai, everybody says okay, it is clearly understood. The homework which I am giving you yesterday, I told you that you have to draw this Krebs cycle. And now I have made it very, very simple for you by giving you two things. The sentence, see, I know some simple formula making options, and this key or important thing which I have written on the side of this sheet. So if you know these two things, nobody can stop you from getting 5 on 5, and nobody can stop you answering questions on Krebs cycle. So if this is Clear? Then we will move to the next thing, and that next thing is energy gain. Or okay, I have done the energy gain here itself, so we can skip this. And now let us come to electron transport chain, the introduction part. Where is what happening? For this, let us draw the mitochondria, and we all know how mitochondria structure is. The outer membrane is smooth, the inner membrane is thrown into folds, right? Let us enlarge this whole thing. Suppose we make a big mitochondria. To understand what is happening in this and what or which reaction takes place where. Let me label this. This is the outer membrane. This is the inner membrane, and we said this outer area, that means the space between outer membrane and inner membrane, is called the outer compartment, or it is also known as perimitochondrial space. Okay, and this inner space is the inner compartment, and it is filled with a jelly-like material which is called the matrix. So this substance which is here is matrix, and this is mitochondria, that means it is in the cytoplasm. So what is here? Cytoplasm. So now, up till now, what we have done is that glucose is broken down into two molecules of pyruvic acid, and the process is known as glycolysis. So where is glycolysis taking place? Glycolysis takes place in cytoplasm. The two pyruvic acid molecules which are formed, these pyruvic acid molecules enter into this outer compartment. The pyruvic acid molecules, two pyruvic acid molecules, change into 2-acetyl coenzyme A. These 2-acetyl coenzyme A's enter into the matrix, and here they are accepted by oxaloacetic acid, and a cycle takes place here. And what is this cycle called? It is called Kreb cycle. So Kreb cycle is taking place in the mitochondria. So now we know where these reactions or these steps taking place. Glycolysis in cytoplasm, this is oxidative decarboxylation, and this is Kreb cycle. So this is Kreb cycle which is taking place. So we know the location of all these things. Now the basic idea of giving you this everything, every information is NADH2 which are formed as a result of glycolysis are here. 2NADH2, and we have ATPs. 2NADH2s are formed in oxidative decarboxylation, and all others which are formed here like 3NADH2, 2FADH2 and 1 ATP, they are found in the matrix. The next step is going to take place, which we call the electron transport chain, is going to take place in the inner mitochondrial membrane. That means here in this part, that is. So this is the place where electron transport chain or this step is going to take place. Now what exactly happens? Suppose I draw, this is the outer membrane and we draw, this is the inner membrane. We are just showing it straight. What is our plasma membrane made up of? It is made up of phospholipids and proteins. Proteins are of two types, extrinsic and intrinsic proteins. Right? Do you know about intrinsic proteins or integral proteins? Did you study the structure of plasma membrane, the fluid mosaic model? So in the plasma membrane or the inner membrane, which is a typical biomembrane, there are some proteins. And when electron passes through these proteins, they act as the transporters of electron. So electron will enter into this, then from here to the next one, then next one, then next one. So it is helping in transport of electrons, and that is why all these proteins which are present in the inner mitochondrial membrane form the electron transport chain. So these electron transport chain proteins are the integral proteins or intrinsic proteins, because there are some proteins on the outer surface of the plasma membrane which are called extrinsic proteins, and these extrinsic proteins, they can act as carrier molecules, so they can move from their position, but the intrinsic or integral proteins, they remain where they are. Okay, so I have given you just a brief idea of what happens in electron transport chain. The electrons are transported through a chain of proteins, and these proteins are in the inner mitochondrial membrane, located in inner mitochondrial membrane. Now, as we have drawn this diagram, I am going to add one more structure, and we will talk about the last step also. On these folds of inner mitochondrial membrane, they are called crista. And on these crista, you find some bulb-like structures, knob-like structures. These knob-like structures, they are called the oxysomes or F0, F1 particles. Oxysomes or F0, F1 particles, or they are also known as elementary particles, and this is the site for oxidative phosphorylation. And what is happening in oxidative phosphorylation is synthesis of ATP. That means this is the place where we will actually get ATP. So now when we go to a bank to deposit a check, so there is a deposit counter where you actually deposit the check, and then there is a cash counter from where you collect the cash. So electron transport chain is like a depositing place where our checks will be deposited, and oxysomes are going to be our cash counters, from where we will be actually collecting or getting the ATP molecules. All right, so now do you have the idea of all 5 steps: glycolysis, link reaction, Krebs cycle, ETC and oxidative, where exactly these steps take place? Is this diagram clear, and is it helping you in understanding the location of all the steps? Hiren says it is good, clear. What about others? Any confusion, any doubt? Kalai says good, it is all clear, very good. So now 5 steps, out of 5 steps, we have done first 3 in detail, and now we have the rough idea of electron transport chain also. Rashmi also says understood, that's very good. Okay, so what we will do is, in the next class we will start with electron transport chain. Now one thing which you have to check before that is, in the new book, NCERT book, there is an electron transport chain given. A diagram is given. Just go through that diagram and try to understand it. There is a text also given next to that diagram. Try to read that text. And then when we meet tomorrow, I'm going to ask you a few basic questions. And then I'm going to explain everything to you. But now I want to know that when you have done 3 steps, are you able to understand the 4th step? The bookish language which is of the 4th step? So this is your homework. Rather, I have given you 2 homeworks. One is practicing Krebs cycle. You will draw that Krebs cycle complete, exactly the way we have done, exactly same. Okay, and second is read that text which is about ETC. And in energy gain, what you have to remember is, from 1 glucose we will get 2 Krebs cycles. Okay, so 1 cycle gives 12 ATPs, so 2 Krebs cycles will give us 24 ATPs total. And this is when you are starting with 1 glucose molecule. How we have these 24 ATPs, that we have talked of. Okay? Alright, now I have some questions for you. You can write these questions down, or you can take a screenshot, and you have to give me the answer of these 4 questions tomorrow. So I'm going to wait. There are some people who are writing these. Kalai says I'll do writing. Okay, very good. So for these 4 questions, they are based on Krebs cycle. So let us see if you are able to solve these 4 questions. I gave you some questions yesterday also. Hiren says screenshot. Whichever way it works. So write down these 4 questions. Tomorrow, the first thing first, we will be doing this. If you have 1 molecule of pyruvic acid, how many ATPs will you get on its complete oxidation? Second, if you have 1 molecule of 3-PGAL, how many ATPs will you get on its complete oxidation? Where are the enzymes of TCA cycle located? And fourth, what is the number of ATPs obtained from 1 NADH2 and 1 FADH2? Very simple questions, but it will help you revise the entire thing. Are you done writing these questions, or have you taken the screenshot? I guess Kalai is writing. Hiren has taken a screenshot. Rashmi did. Okay, very good. You wrote those questions down. Try to solve them and we will answer these questions tomorrow. First thing first. And then we will start with electron transport chain and oxidative phosphorylation. So is the Krebs cycle clear? How confident are you on Krebs cycle? If there is any question asked on Krebs cycle, will you be able to answer it confidently and correctly? Oh, Hiren says 101%. That's very good. What about Kalai, Rashmi? How confident are you now? Okay, thanks Rashmi. I'm so happy you liked it. Kalai has also written the questions. Okay, so I guess everybody has understood. Kalai also 100%. That's very good. So one major step in aerobic respiration is done, that is Krebs cycle. Now when we meet tomorrow, we will start with these questions. We'll go over it. And I'm 100% sure that all of you are going to get the correct answers, but still I'll go over it, and then we'll do electron transport chain and then oxidative phosphorylation. All right, so I'll see you tomorrow, and don't forget to take the subscription, and don't forget to use this code, because this code is going to give you that 10% discount. So if you have not taken the subscription, take the subscription as early as you can. All right, okay, so I'll see you tomorrow. Till that time, don't forget to finish the homework. Okay, bye.